Good morning, everyone. I am Hari, and it is my pleasure now to introduce our keynote speaker, Professor Dr. Iain Cheeseman. Iain is a member of the Whitehead Institute, along with a professor of biology and associate department head at MIT. He earned his PhD in molecular cell biology from the University of California, Berkeley in 2002, following which he conducted his postdoctoral research with Arshad Desai at the Ludwig Institute and University of California, San Diego. Iain's lab investigates the mechanisms regulating cell division and chromosome segregation in different model systems. His lab has discovered and characterized numerous components of kinetochore, a macromolecular structure vital for chromosome attachment and movement during cell division. His lab is currently interested in how the cell division machinery adapts under various physiological conditions such as development, aging, and disease. Iain's contribution to the field have been recognized through numerous prestigious awards, including the American Society for Cell Biology's Earlier Career Life Scientist Award in 2012. He was also Keith R. Porter Fellow in 2013 and 2019 and was named as an outstanding mentor by the MIT Undergraduate Research Opportunities Program. So please join me in welcoming Dr. Iain Cheeseman.
First fail. I lost my package. Thank you very much. Thank you. It's really good. Can everybody hear me? Okay? Yeah. It's great to be here. I really appreciate the organizers for doing this and Hari for everything for just getting us all here. I'm looking forward to the next few days as well, and I'm particularly excited to be able to tell you about the recent work from our lab. So I'm going to tell you some two recent stories on this question of rewiring cell division. But one thing I'm going to try a little bit different for this talk, it's a new experiment, you're going to have to bear with me for this, is that I'm going to give you a little bit on the side about sort of how I think about science too. So several years ago, actually, when Twitter was still a thing, I put a thread up there sort of describing my thoughts and ideas about how science ought to work. And so I'm going to sort of, that thread is way too long. I encourage you, if you're interested, you should check it out. But I'm going to take a few of these and sort of intersperse them throughout this talk as well, okay? And in particular, I think that the science that we do, the things that we discover matter. But in my opinion, the way that we do science also really matters. And I think that this sort of, student and postdoc organized symposiums are a really great way to bring this. And so I think just as the broader statement, if there's nothing else that you take away from today's talk, do science in a way that's positive. Each of us have that ability, and we're empowered to make a positive difference, and you have that within your control. And so I really encourage you to find the good ways to do that, and even just the way that you interact and treat other people matters. But I think that everything that each of us does has the ability to make a community better. So I'm going to bring that in throughout, but I think particularly what I'm going to tell you about today is our work on cell division and particularly the sort of molecular machines that comprise a cell. So I love this picture. Actually, I took this picture when I was a postdoc in Arshad Desai's lab. I think for me, I've looked at this picture probably 1,000 times, and every time I see something slightly different. In particular, so here we're seeing a cell in mitosis. The DNA is in blue. The microtubules are in green. These provide the structure and force for cell division. And these red dots are the kinetochore. You look at this picture, there's a lot going on. But for me, what I see is the hundreds of individual molecular machines that are comprising the spindle, the kinetochore, the chromosomes, that have to work individually and in combination to achieve this. And so I really like thinking about these machines and the molecular players there. What I think is beautiful about these machines, too, is that they're not just simply static machines. They're actually quite flexible. And this is really going to be what I'm going to focus on today, is this idea that the cell builds machines, but these machines need to be able to be tuned and adapted and altered across a range of different circumstances. And so I think it's one thing if we imagine a HeLa cell, for example, at this one stage in mitosis. But if we think about the diversity of cell division, there's a lot of different circumstances under which cells find themselves. So this is a picture of a multicellular starfish embryo, actually, or early cell divisions. And I think here, thinking about just how different these rapid early embryonic divisions are, really can start to make you think about why you might need to change these processes. So across the context of our lab, we're actually interested in a range of ways in which cells adapt and adopt their behavior. And so mitotic division is one thing, but if you think about how that changes to make the gametes during meiosis, how this is altered over the cell cycle and across different cell states like quiescence and senescence, across tissue types, for example, and then also particularly in disease, so aging, cancer, and other sort of rare diseases. There's a lot of changes to the different cell division processes. So I'm going to talk about this rewiring today. What I'm not going to have time to speak about is that our lab is also quite interested in taking these approaches to a large scale. And so a lot of the work that we've been doing has used optical pooled screening and other functional genetic strategies to really scale the way that we're doing cell biology. Okay. So I mentioned I was going to intersperse some things. And I think one thing to describe about myself is actually I'm a very emotional scientist, in the sense that when I've done stuff that I did because I thought it was the right thing to do or that was savvy in some way, it's never worked out. But I think that when I've done science that makes me happy, it works out a lot better. And so I think what I'm going to tell you about today is the science that makes me happy, and some recent work from our lab that I've just really enjoyed living vicariously through, from a postdoc, Mary Jane, in our lab, and a grad student, Jimmy Lee. And I do think that I encourage each of you as well to find those things that you like. We tend to think about there's something that's hot that we really ought to work on. Find the thing that makes you happy, and I guarantee you're going to be able to convince other people that it's also something that you should focus on. Okay, so before I do that, I'm going to try to answer one question, the thing that really should have been the question that all of you had when you came in here, which is my title, Rewiring Cell Division. What the heck does that mean? And so I'm going to show you a brief animation that sort of explains how we got to this point and why this is sort of the theme in our lab, okay? And I'm going to do that by starting with this Western blot here, okay? So here we have a Western blot. This is a protein that was discovered more than 30 years ago. It is something for which since then there's been about 1,300 papers on. And this is a Western blot for this protein. Actually, it's a commercial antibody, a lot like other things that have been done in the previous on this protein. In this room, can we just have like brief show of hands of who's done a Western blot before? Okay, good, like maybe 80%, that's good. Okay, this is what this Western blot looks like. You know it's not what the Western blot looked like. Okay, so the Western blot is much more complicated than that, and we can see the canonical protein here. We simplified things. I wanted to make it a little bit easier for you to see what that is, so we cut it out. You probably did that for your papers too, but you know that there's other stuff on that. Okay, and so when we look at these bands here, our default assumption is that they're antibody-specific background. So antibodies are great, but the chance of them recognizing an epitope on another protein is legitimate, and so clearly what these are here is background bands from this Western blot. Okay, but we can do a better experiment, and particularly Mary Jane, who did this work, was like, okay, well, let's do a slightly better experiment. I can get rid of the mRNA that's coding for these by RNAi, and then I can look at, okay, what bands remain. And so our assumption is going to be that this canonical protein is going to go away because we got rid of its mRNA, but these non-specific background bands are going to stay. And so when we do that and Mary Jane depletes the RNA, you can see that the canonical protein got eliminated and this band stays. This is clearly antibody-specific background, but we see loss of these other two products as well, okay? And so here now we have sort of a second default assumption, which is that actually what these represent is breakdown products. Okay, so Mary Jane is the most careful scientist I've ever met in my life, but maybe when she went in to do this, the conditions she used to break up that cell extract resulted in sort of, it sort of being broken down and proteolytically cleaved and things like that too. And so what these represent is breakdown products, okay? So we can do a slightly better experiment now where Mary Jane can go in and just actually get rid of the start codon for the gene. And when she does this, you lose that canonical protein, but these other two proteins remain, okay? And I think for us, this moment was kind of shocking to think about what we had been ignoring. So this gene, which I'm going to spend a lot of time focusing on in just a second, it's only a single mRNA. Again, a lot of work is done on this. Everybody else's Western blots also had these products, and they just really ignored them. And so I think that a lot of what we're doing is we're looking really above the surface. When we think about using a Western blot or even mass spec with a defined protein database, we have a lot of core assumptions about what these molecular machines are, but there's a lot hiding under the surface. And so I think for us, we're really interested in understanding how molecular machines can be flexible in this way through these kind of alternative protein products. So when we think about the central dogma that all of us learned very early on, our general assumption is that a single piece of DNA is going to code for a single protein. We know that's not true. And we know instead that it's possible for a single piece of DNA to make many different protein products. And really about every single step along this process, and in which there are many, can be altered and adapted in very specific ways. So for example, we can have alternative transcription start sites, we can have post-translational modifications which will functionally change that protein, proteolytic clock processing, alternative splicing, alternative translation initiation, and many of these other things can generate a broad diversity of protein products. And it's this potential to make these products, but to also shape how these products exist across different physiological circumstances. To me, that really is what rewiring means, that ability to sort of alter and change things over the course of different circumstances. And so I'm going to show you some examples of that today. So the process that I'm going to primarily focus on is mitosis and cell division, but particularly when cell division is challenged. So a core part of the logic of cell division, both in physiological circumstances or in cases that we're trying to sort of target cancer cells with chemotherapeutics, is that when there is some kind of damage, this leads to a mitotic delay, okay? And really what the cells want to do is they want to fix this damage and so that they can correct their chromosome alignment, and then they want to be able to divide, okay? But under circumstances under which that damage is persistent, both within our bodies naturally or because of chemotherapeutic, at some frequency the cells can actually exit mitosis prematurely through a process called mitotic slippage. So these are arrested cells in mitosis, they're rounded up, but if you watch this one, what's going to happen here is that's going to now exit into a sort of flattened G1 state, okay? And so those two cells, as you watch them there, it's not that they fixed this and they're damaged and divided. They just decided that they were done with this and that they were going to revert to a G1 state. So they did not divide. They did not segregate their chromosomes. They're now a tetraploid G1 cell. It's not ideal, but if you're a cancer cell and actually many other cells in our body, it's actually a better thing to be a tetraploid cell and have another attempt to do cell division than just end up going through apoptosis, okay? So I find this process really fascinating, but this timing here, if you watch that, they hung out for a while and at some point they decided to go. And so actually there's a pretty much of a set time for a given cell line for how long it's going to do that, but that process itself is dramatically variable across cells and cell types and cell state. And so I've always really been curious about what that logic is that controls how long a cell is going to stay arrested in mitosis before it proceeds. And we want to understand this because we want to understand it. It's a fascinating cell biological process, but also we need to really understand this if we want to think about how we treat cancer cells, for example, with Taxol. So if we start to think about the molecular machines, there's a lot that we already know about what those molecular machines are. And key amongst these is an E3 ubiquitin ligase called the anaphase promoting complex with a key adapter protein called CDC20. And this machine is really central to a lot of the regulation that happens at mitotic exit. So this E3 ligase degrades proteins that are required for sister chromatid separation. We're going to hear a lot about genome organization, things like that today too. And it's also, its job is to promote mitotic exit, particularly through degrading cyclin B. Okay? And so it functions as a great machine to be able to drive these forward. And when the cell is happy, it's got attached kinetochores, it's going to do that. But in the presence of some sort of damage, there's a signaling pathway called the spindle assembly checkpoint, who basically, and there's a lot of details here, you don't need to remember any of them, I just want you to remember actually this one protein CDC20. But the job of this signaling pathway really is to come in and inhibit this E3 ligase by binding to CDC20. And in this case, this is going to block mitotic progression by just inhibiting CDC20. It can't act. Okay? So we know a lot about this molecular machinery. We really don't know how this machinery is tuned and adapted across different circumstances to allow these different behaviors and outcomes for cells. Okay? And so our surprise came when Mary Jane was looking to do functional genetics to try to understand how CDC20 worked. So our expectation was that if you get rid of CDC20, because it is the factor that drives mitotic exit, if you don't have it, the cells are going to rest in mitosis. And in fact, if you deplete CDC20 by RNAi, or if you use CRISPR-Cas9 to cut a region here, generate frame shifts and sort of non-functional protein, even at early time points, the percent of mitotic cells is going to start to increase. And so it really is arresting the cell in this way. So the process works well to deplete it. But for us, actually, these came a little bit later. And so the first guides that Mary Jane made to target CDC20 were a little bit earlier. And when she did this, she saw nothing, okay? And so in this case, these cells were not arresting in mitosis. They didn't really have any discernible phenotype that we could, okay? This was kind of a shock for us because, this is not the way CC20 should work. And so actually, for about 2 years, we were in this mode of, we are doing something wrong. There's a problem with things technically. We are just not good at CRISPR, even though at that point we probably targeted more than 300 genes. There was something technical that was a problem. And I really want to highlight here, both for this and pretty much everything I'm about to show you, Mary Jane did just such a wonderful job of walking through completely unexpected behavior and trying to understand how things work. And I really encourage you, for each of the things that you're doing, figure out how things work. We each have a model, we have a hypothesis, there's dogma in a field, but your job is not to prove that model, your job is to really figure out how things work. And so for the things I'm going to tell you here, actually Mary Jane's goal was not to prove something, she spent every single step along the way trying to prove us wrong. And so some of the things that I'm about to show you were really quite unexpected for us, and so she came up with every alternative explanation that she could along the way, okay? So the logic here, again, is that these early guides didn't have an effect, and so it certainly could have been technical. But as hopefully starts to feel a little bit familiar now, this is that protein that I showed you about on the Western blot. And so normally CDC20 has this large canonical protein that people have been studying for years, but if you get rid of the start codons or you make internal frame shifts, you can see that there's clearly these other two protein products that are here. And these are really the explanation for this. But in these knockouts, even though CDC20 is an essential gene, only one mRNA that's producing it, what Mary Jane was able to find is that these cell lines were completely happily behaved. So in this mutant, for example, this M1 stop mutant here, you can see that its growth curve is virtually identical to control HeLa cells, okay? And so even though you need this full-length protein, it seems to be, even though this full-length protein exists, it seems to be dispensable for viability because of these alternative protein products. So what are these different protein products? So I'm sure you can see this really well in the back of the room. This is actually the N-terminus of CDC20. It goes on, it's about a 450 amino acid protein. So you were just seeing the first 100 amino acids here. But if you gaze at that sequence carefully, what you can see is that there's 2 additional methionine residues in the first 100 amino acids. And the size of the products that would be generated by the use of these sites, it was actually quite consistent with those other bands. And so if we think about just the way that translation works, so here we have the mRNA sequence for CDC20. Normally a ribosome is going to be loaded on the cap, and its job is to scan along until it finds that first methionine where the 60S joins, and now we're going to make a protein product. But the logic of translational control in actually all organisms, but particularly eukaryotes, is that this process is leaky. And so this other ribosome here can scan along, maybe it sees that, but just sort of ignores it, and it can go and it can be able to begin translation here instead, okay? And so actually many of our genes have this kind of decoding where we take it for granted that there is a specific start site that is used, but alternative start sites, including downstream start sites, can also be used with high efficiency. In this case, Mary Jane was actually really able to quite nicely demonstrate that this was happening. So for example, she tagged the C-terminus of the endogenous locus for CC20, and when she isolates down that protein, we can actually not only get peptides corresponding to this first methionine with a cleavage and acetylation event, but we can also see this, both of these other methionines there. And so these peptides really would never exist unless they were being used as alternative start sites. So we're really quite confident that they are being used in that way. And then I think probably most impressively, she was able to sort of reconstitute this. So here we have an experiment. We're showing a Western blot. This is a cell in which Mary Jane has depleted all CDC20. You can really see nothing there. And then she can build this back by just adding in a cDNA. And so even though we knew that this was not alternatively spliced within the coding sequence, this was a really nice proof that just this ectopically expressed cDNA could recapitulate this banding pattern. And from there, she could go through and walk and eliminate each of these individual downstream start sites by mutating them to leucine. She can get rid of the start codon. The nature of leaky scanning means that you should even see more of the downstream products, which we do, and then we can eliminate those again as well, okay? And so we're quite confident that we have a situation now in which cells are actually always making more than one version of this protein product, the full-length version, but these two shorter versions as well. So how are these different? So if we come back to the sequence, we have the canonical start site, we have a downstream start site, and so both of these alternative products down here are going to essentially miss this region up in the front. And if we look in that region, one of the things that was known from prior work, particularly from Hongtao Yu, Andrea Musacchio, and Arshad Desai, and a few others, was that this region was participating in the formation of that inhibitory complex I highlighted for the spindle assembly checkpoint. And so some of the predictions is that if you lack this region, it should change the way that CDC20 is behaving with respect to its checkpoint control. So what I've already showed you is that mitotic progression is fine. Those cells keep going through mitosis, but does this change the way that they're able to be inhibited by the spindle assembly checkpoint? So in this experiment here, I'm showing you sort of a mitotic timing survival curve. This top line here is control cells. They've added an antimitotic called STLC. It sort of blocks spindle assembly. And here we're just basically seeing the intermitosis and these cells are staying. And actually HeLa cells will arrest in mitosis for about 1,800 minutes. And so this curve is kind of going way back over there. Okay, so they will stay arrested for a long time. In contrast, if we get rid of that full-length CDC20 protein product, these cells will immediately exit mitosis. And so they really functionally lack this spindle assembly checkpoint. So we have a nicely inhibited full-length version and a completely uninhibitable short version. Okay. So just to summarize what that looks like, if we see here, some sort of mitotic damage, normally this is going to activate the spindle assembly checkpoint to inhibit CDC20, and that's going to block its activity, leading to a mitotic arrest. In contrast, under the circumstances where Mary Jane made the cell only have the short version, this is going to really prevent these from inhibiting CDC20, and this is going to allow a mitotic exit even if you have this damage. Okay? So we have these two very different circumstances, product A versus product B, but I think that actually the most fascinating thing about this, and the most fascinating thing for thinking about this ability to rewire these core cellular processes, is that actually cells simultaneously have both of these. And so what Mary Jane's hypothesized is that actually it's the balance between these that really is setting and changing the way that the cell behaves. So you can imagine, for example, if you increase the full length, maybe more mitotic arrest, you increase the short version, maybe more mitotic exit, and it's the balance of these that could really be creating this timer for when a cell is going to slip out of mitosis. Okay? So Mary Jane wanted to test this model. And so we can actually do that in sort of subtle ways by actually just tuning the ratio and balance of these. So if you just tune up the full length in a slight way, and so she did that by essentially getting rid of these two downstream start codons in a partial replacement assay. So it actually works pretty good, but it's really not complete. There's still some of these small protein products. We can actually look at what happens, and our prediction is that this should increase the duration of the mitotic arrest. And that's exactly what she saw. So on this graph here, each one of those dots is an individual cell. It's how long that's spent in mitosis. On average, HeLa cells spend around 1,800 minutes in mitosis. But you can see there's some spread, but a pretty good timer. But if just by increasing that level of the full length, now we move up to an average of about 2,100 minutes. And everything in black actually is still in mitosis at the end of the movie. And so this is underestimating how long they'll arrest. Reciprocally, Mary Jane can just tune up the levels of these short isoforms, and she did that by messing with the alternate translational control. I'm not going to go through the strategy so much, but you can see that now there's an increased amount of these lower protein products. And in that case, just a small difference, again, goes from about 1,800 minutes now down to about 1,400 minutes. And so we can really shape this timer, okay? And encourage you to check out Mary Jane's paper, which is now published. She played a lot of tricks like this. We're very confident that this provides a really elegant timer in mitosis, and it's through a combination of ongoing synthesis and degradation. But you can really define a timer just simply by the turnover of these different isoforms. Okay. The last part of this that I am going to tell you about, though, is what I said at the beginning, which is I think that this matters not only for the way that we think about regulatory control, but the way that we think about cancers. And so Taxol, paclitaxel, really is a frontline chemotherapeutic for a number of cancers, including lung cancer, breast cancer, ovarian cancer. And so we were really interested in understanding the contributions of this kind of behavior to the way that cancer cells behave. And so one thing that Mary Jane noticed actually is that there's a number of mutations in CDC20 across cancers and tumors that would be expected to specifically affect these alternative isoforms. So here, for example, this is a premature stop codon. And in this case, actually, this endometrial cancer there, we were able to get this cell line, which is a heterozygous mutation that creates a premature stop codon CC20. If the full length was the only version, this should be quite bad. But in this case, our prediction would be that this would create a higher amount of these downstream products. And that's exactly what she sees. And so HeLa cells have these smaller products, but this HEX6 cell line has a very much larger amount of these downstream products. And this HEX6 cell line, actually, it can only last in mitosis for about 4 to 5 hours. So it really correlates very closely with these behaviors. So we're seeing cancer mutations that would be specific to predicted to affect this. We're actually seeing changes in these isoform ratios that have nothing to do with the underlying coding sequence of CDC20. And so here, for example, if we look at HeLa cells under the short exposure conditions, you really don't see these alternative protein products. But U2S cells, which is quite aggressive osteosarcoma, have much larger amounts of these downstream products, and then also actually spend much less time in mitosis, so a correlation. I'm certainly not saying this is the only difference between these cell lines. Of course, they're cancers, there's a lot of differences, but we think that at least in part, these isoform usage can really explain that. Okay. So we are seeing some evidence that cancer cells are adapting this. Is there consequences of that to the way that we treat cells? And so recent work from our lab has used a lung cancer model, A549. I know it's actually one of the posters today from Varoslav also is using this A549 cell line. It is a great lung cancer model for looking at tumorigenesis. And in these xenograft assays, if you treat with paclitaxel, it will do a good job of hitting the control cells. It's actually a relatively small tumor, but you can see the control cells are still forming tumors under this dosage, which is quite physiological. But actually, if we take a version which is lacking the full-length CC20, these tumors are exquisitely sensitive to paclitaxel treatment. And so just getting rid of this checkpoint control really is having a consequence. This, actually, we were hoping for, but we're quite pleased by that this is synergizing for the treatment. What we didn't expect is that in control cells, the A549, this is not to scale, form quite large tumors, but the mutant, CDC20 mutant lacking the full-length isoform is an even more aggressive cancer. And honestly, we really didn't believe that this was the case. Okay, maybe we just injected too much. And so we went to the place where we could actually use an in vivo competition assay. We just had these cancers race. So we co-injected equal amounts of control cells and these mutant cells lacking this full-length product. And when we do that, what we see, and this is work from Sarah Katie in our lab, what we do that is that compared to the initial injection, the mutant is actually much more aggressive. It will outcompete the control cells, and yet is exquisitely sensitive to the paclitaxel. And even low doses, you can actually get rid of these cells almost entirely. And so moving forward, we're quite interested in sort of leveraging these findings to think about the way that we would diagnose and treat cancers. Okay. So I'm going to transition now, and I think hopefully this one story has sort of shown you why we have this strong love for this concept of rewiring, this concept that we're building molecular machines, but these machines are flexible. And so just in the case of CDC20, the alternative translation start site there is allowing a single cell to create multiple protein products and to be able to shift and alter the balance of these protein products over time to influence and alter the behavior of that cell, okay? And so we had this result from Mary Jane, and we're like, okay, this sort of changed our world. We really cannot understand the logic of mitotic arrest duration unless we knew about the existence of these isoforms. There has to be other things like that too, okay? And so we were interested in trying to understand, particularly on this level of alternative translation initiation, are there other protein players that would be acting in the same way, okay? And so there's got to be at least one other one out there, okay? So for this second part of the talk, the work I'm going to tell you about is from Jimmy Lee, who's a grad student in our lab. And Jimmy Lee was really interested in understanding more broadly the factors that are reshaping sort of the way that the proteome is produced. And for this, I think mitosis is a really interesting place, but it's something that honestly everybody ignores from the standpoint of the central dogma. So I think that partly your assumption is that mitosis is a silent place. There's not a lot going on there. There is not new ongoing transcription during mitosis, and so we really shouldn't care. So I'm going to show you something that actually really surprised me for this. So here is an experiment that Jimmy did where he looked at cyclohexamide addition to block new protein synthesis. He did the same thing with puromycin. And actually, if you treat cells with a translational inhibitor, they're fine, okay? And I think many of you probably have used puromycin to select for cells. It worked somewhat quickly, but you know it was a few days before they ended up dying and before you were able to select the cells. And in fact, so at 8 hours, you don't see anything. 24 hours, modest cell death based on propylium iodine staining. It's not really until about 2 days that you're going to see the cells starting to die just by blocking a new translation, okay? Mitotic cells are strikingly different. So again, part of the physiological context of a mitotic cell is that when there is some sort of damage, it wants to be able to delay that. And so if you take that delayed case and you ask whether new translation is needed, with as little as 4 hours of cyclohexamide addition, these cells are going to begin to die, okay? So ongoing translation is absolutely needed during mitosis, and not only is it happening, but the cell absolutely depends on that for its viability. And so Jimmy's like, okay, we really need to understand not just what's being translated here, but like exactly what is changing, what is different, what does mitotic translation look like? So Jimmy went through and he tried to systematically define the nature of the transcriptome and the translatome. So he took interphase cells that are growing without mitosis, and he took cells either arrested or synchronized into mitosis in various ways. And he went through and he generated, did RNA-seq to look at the transcriptome, standard ribosome profiling, but then also particularly start site ribosome profiling. So using a combination of drugs that we can block the initiating ribosome on the translation start site, and asked where are those and how does that change? Okay. So I want to think about the protein products that you could actually get from this, and particularly from the ribosome start site profiling. So the standard that we're going to get is an annotated ORF, and just to say, so I'm really proud of Jimmy's data set, but just to highlight, a large number of labs have conducted similar studies in asynchronous cells for looking at these products. But of course we know the annotated ORF. These are the things that each of us are predicting computationally. There's also the potential for these N-terminal truncations because of the leaky scanning, the things that I just showed you for CC20, where it's going to ignore that first product and go downstream. And then I think the ones that are not really on most of our radar is the potential for a in-frame translational extension. And so one of the reasons that these exist is that although we assume that AUG is the start codon, this is start codon, right? But actually near cognate start codon, CUG, GUG, ACG, UUG, can actually also quite readily be used as start codons under various circumstances. And so the Kozak consensus sequence that was really defined a while ago by Marilyn Kozak really ultimately is a statistical likelihood of a start site being used. And so although AUGs tend to be the best with the right context, many other sites can be used too. And in this case, you can generate these a longer protein than you expect. And then of course, there's also UORFs up there, many of which can be regulatory. So when Jimmy did this in HeLa cells, he was able to identify more than 7,000 annotated start sites. We're pretty confident that we're doing a good job of this. So I would guess that in a HeLa cell, there's probably about 10,000 genes that are expressed. And so we're capturing a large portion of these. But what really surprised us is that what Jimmy was also able to see is more than 5,000 of these flavors here. So more than 3,000 translational extensions, more than 2,000 truncations. And so I can pretty much guarantee that each of you thinking in your head, whatever your favorite protein is right now, if you have more than one of those, one of them likely has an alternative protein product, okay? And so what I showed you for CC20 is not the exception. I think it really is typifying the rule, that there's a huge diversity of translational control that's out there, and it really has the strong potential to shape a lot of that biology. The other thing that really blew us away from Jimmy's work is just how dramatically the use of those start sites change over the cell cycle. So if we just look globally at all these translation initiation sites and we compare an interface cell to a mitotic cell, there is a massive rewiring of the usage of this during mitosis, with thousands of sites actually increasing in their usage during mitosis and thousands of sites decreasing in their usage, okay? And so when Jimmy looked at these and tried to understand what was on the left versus on the right, the thing that really stood out to him is that the canonical AUG start codons were preferentially used in mitosis, but these non-canonical start codons that I just mentioned, like CUG, GUG, are actually repressed generally during mitosis. Not in every case, but in many, many cases. Okay? So we can look at this here and just, average efficiency in mitosis AUG is quite good. These other non-canonical start sites are down. And I should just point out, I'm not going to go through this, but poor Kozak consensus AUGs are actually also down. And so it's not just the start codon entity, but really this sort of efficiency of the start site that's changed. So we can see this in ribosome profiling. Jimmy can actually also recapitulate this in a number of ways. The one that I'm showing you here is a luciferase reporter that basically is comparing AUG to CUG translation. During mitosis, the AUG is modestly repressed, and there is a translational attenuation overall, but still active translation. But the CUG is dramatically repressed, so it's about a 10-fold repression compared to interface. And so we're really quite confident that there is a change in the sort of relative usage of these start sites. Okay. So we know that there's this big reshaping of the proteome through the alternative control of these start sites, but why is this changing? What's actually different between these states? And so just to highlight, this is a really rapid and switch-like mechanism. The interface cells really behave one way. The moment that you are in mitosis, you switch to this much more stringent translational control, and then as soon as you exit into G1, it gets back to that original place. And so why is this change happening in this dramatic way? So Jimmy decided to go to the source, which is that he wanted to look at the ribosomes, and particularly how translation initiation factors associate with them. And so we isolated different fractions of ribosomes, including the 40S from polysome fraction, and compared them using quantitative mass spectrometry within our lab. And so when you do this, actually most things in the 40S fraction are largely unchanged, and this is the 40S subunits themselves, as well as a number of the standard translation initiation factors. But you can see that there's a few outliers. And particularly what Jimmy found is that there's a strong increase in EIF1 associations during mitosis, and a strong decrease in EIF5 associations. And so when he found this, I got kind of excited that there was a difference, but for Jimmy, who knows much more about translation than I did when he started this project, it kind of explained everything, because these have a very well-established role in controlling the efficiency of start site usage. And so particularly EIF1, when its levels are high, does a really good job of promoting strong context AUGs, but repressing poor context AUG and non-canonical start codons, where EIF5 really has a competing effect. And so its job is to essentially promote these more lenient usage of what start sites to use. And so what I showed you is that there's much more EIF1 bound to the ribosome, really would explain what Jimmy is seeing in that behavior. And so there is some change that's resulting in a difference in EIF1 and EIF5 associations. What is that change? And that's ultimately what we need to understand. And so Jimmy actually considered a variety of explanations for this. The first one was mitosis. It's got to be phosphorylation. But we cannot find any evidence for mitotic phosphorylation of either of these proteins or the ribosome itself. Second explanation has got to be protein levels, but we see no difference in EIF1 or EIF5 levels by quantitative mass spec, and just showing you here by a Western. If anything, less EIF1, which is what you wouldn't expect. And so there has to be a different explanation for why there's this massive change in translational control during mitosis. So we're ultimately cell biologists. I love looking at cells. I began with that first picture of a mitotic cell dividing. It's just such a beautiful thing to look at. And so Jimmy said, okay, I'm just going to look at these proteins. And so if you look at EIF5, and this is actually exactly where you'd expect it to be. The ribosome is in the cytoplasm. That's where the translation initiation factor should be. Makes a lot of sense. But EIF1 behaves differently. And so certainly a good population of that is in the cytoplasm, where it can act on the ribosomes. But there's a second population in the nucleus here. And during interphase, that population really shouldn't be able to do much. It can't touch the ribosome. It's not like it has an ability to come in and influence them. And so, yet thinking about the nature of that population, mitosis is going to be a great place to take advantage of it. So for example, if we just look at a nuclear localized version of EIF1, you can see great nuclear localization. The minute the cell is going to enter mitosis, the nuclear envelope is going to break down, and that's now going to release EIF1 into the cytoplasm. And so you're essentially dumping this extra payload of EIF1 where it can go influence ribosome behavior. And then as soon as the nuclear envelope begins to reform in mitosis, it's going to pump EIF1 back in, making this pool anew. So the model, really to highlight the way we were thinking about this, during interphase, EIF1 is in the cytoplasm, but there's this large pool in the nucleus that's excluded. And so this is allowing both use of both strong and weak initiation sites, allowing the cell to really make a wide range of protein products. But at mitosis, nuclear envelope breakdown is going to release this extra pool of EIF1, really serving to repress these weak initiation sites and encourage the use of the strong initiation sites. Okay. So we want to test that model. Okay, so we love this as an idea. What would happen then if you were to eliminate EIF1 from this nuclear population? So Jimmy tried a lot of things, but ultimately the one that I think worked the best is what I'm going to show here, which is able to tag both copies of the endogenous EIF1 locus in HeLa cells. Normally this works quite well, and again you see both this nuclear and cytoplasmic pool. And just to highlight, this is broadly conserved. All mammals have this, all cell types have this. This is something that we're really seeing across a wide range of things. But what he could do is actually create a nuclear export signal in EIF1. And because of a really beautiful feedback loop for EIF1, what this does is basically keep cytoplasmic levels unchanged, but precisely gets rid of the nuclear version. And when he does that, instead of this dramatic decrease in CUG usage in mitosis, now essentially keeps the mitotic state the exact same as the interphase state in terms of start site choice. Okay. These cells are completely fine. They grow completely normally until you challenge them and delay them in mitosis. And then that becomes a problem. And so, as I mentioned again, the logic of translational control, normally mitosis is about 45 minutes. You probably don't need a lot of new synthesis there. But a mitotic cell cannot persist in mitosis more than a few hours without ongoing translation. And in this NES fusion here, again, this EIF1, these cells die within a few hours. This is viability staining. There's about 24 hours here, but Jimmy's done the same thing with an 8-hour version. And also you're getting a strong and dramatic increase in the frequency of cell death. And so under normal unperturbed conditions, you don't need this, but any time that you have mitotic delay, you definitely need this. So what's responsible for this death? So one of the things that Jimmy looked at is if you look just across these thousands of start sites, who is being changed during mitosis, one of the things that really stood out was the mitochondria. And the mitochondria is kind of like the small little organism living inside of us. It's doing its own thing. It doesn't really care if the cell is in mitosis. It still needs to be able to grow, replicate, and divide and do its other stuff. And so there's a strong need for ongoing mitochondrial translation. And so our prediction would be, because of their strong emphasis of these start sites, is that in this mutant, without nuclear EIF1, this process would be compromised. So normally during interphase in both of those different cell lines, mitochondrial production is fine. This is a stain for mitochondrial efficiency. But now if you look in mitosis, these mutant cells really lack mitochondrial function. And so we think this is one of the things that's contributing to their poor viability. In addition to that viability, we see a big change in mitotic progression. So I mentioned when I talked about Mary Jane's work that mitotic slippage is actually quite efficient in many cell lines. And so these HeLa cells that we're using here will undergo a high fraction of mitotic slippage ultimately. But these mutant cells actually really display a much lower percentage of mitotic slippage. They're really not doing that. And so just to come back in full circle, this CDC20 that's being made in these mutant cell lines, you just don't see those smaller isoforms. And so they're really not giving you that normal release valve that allows you to exit mitosis even in the presence of the drugs. Okay. So we're seeing really a broad rewiring of the translation program in mitosis. And again, you think about each of those machines that's being made. It's just not one protein that's being made with these alternative isoforms. there's hundreds and thousands of proteins in which the usage and production of these is going to be shaped in various ways. And so I think for our work, we're quite excited for what Jimmy defined for this level of regulatory control. But in general, thinking about the diversity of protein products that are out there is something we're really interested in exploring further. Okay, so I'm going to give you one final teaser for that, and this idea of not only rewiring cell division, but rewiring many aspects of cell biology. And of the 5,000 genes that I showed you, in which there's a trend of translational extension or truncation, what is that biology doing? Okay. So, and this is now, again, unpublished work from Jimmy. Here we have a tRNA processing enzyme called tRNT1. And we're just seeing here the traces from Jimmy's ribosome profiling data. And so you can see the start site usage here. This first methionine is quite efficiently used. But this single mRNA, this methionine 30 is used just as equally. And so here, this protein is making two alternative flavors in the same cells, A and B. One is about 30 amino acids longer than the other, okay? And I love just thinking about this 30 amino acids and all of the information that could be encoded within that 30 amino acids, all the things that these two different products could be doing. So in this case, what Jimmy did is he tagged this at its C terminus with GFP and just looked at what happened with that native mRNA. And so when we do this, you can see beautiful localization. And this protein, you see really nice mitochondrial localization. That's what those little fibers are, and great nuclear localization. But honestly, this is a little weird. Like, those are two very, very different places in the cell, very different signaling and sorting that goes into putting them there. And in fact, you can deconvolve this. So if you express just this shorter M30 to the end version, only localizes to the nucleus. And if you get rid of that alternative start codon there, that methionine 30, only to the mitochondria. And so what the cell has done is it needs this tRNA processing enzyme both in the mitochondria and in the nucleus. And instead of make two enzymes or do gene duplication, it's given the single mRNA the capacity to make both, okay? We give viruses a lot of credit for how well they use their genomes and being able to make a lot of different protein products. Our genomes are doing the same. We're making a lot of different protein products that are out of there. And I think that there's a series of biology to understand out of this, but also it impacts disease. And so actually this mutation here, this M30V mutation, ends up being a patient mutation that results in a B cell immunodeficiency. So multiple patient families actually have an M30V mutation. You would ignore this, it's a conserved hydrophobic, unless you knew that that was an alternative start site. And so we're really interested in understanding and unlocking that potential too. Okay. So to thank people who contributed to the work, so Mary Jane was a former postdoc in the lab who's now working in Australia. Jimmy Lee is the grad student who drove forward the second half of the work that I said. And this is the rest of our lab. I also mentioned work from Sarah Katie. One thing I just want to add, coming back to what I said at the beginning, I love this group of people. And I think that I told you work about what Jimmy did, but like science cannot occur in the vacuum, okay? And I think that just the way that we can build synergies and support each other, and particularly it's very important to me in the context of our lab that people do that. We're really fortunate to benefit from great staff support too. And then I think really generally, we're all on the same team. And genuinely, I mean this for our lab, that our efforts benefit each other and make a difference. But actually for all of us and all of us in the room, the things that each of you all will discover, the things that you're going to do, the things we'll see at the posters today, those are great for me. Those are the discoveries that are going to drive forward our own science and I can live vicariously through. We can all benefit each other by the way we think about supporting each other and interacting. And I'm going to leave it there. I'm going to put a few one up that I'm not going to read through in this way. I told you it's a very long tweet thread, but I think that I'm going to leave that there for the 10 minutes of questions or whatever. And I'm over the next few days, I'm really looking forward to chatting with you all about your careers and your science and how you navigate things too.